Good evening, everyone. Welcome back to the July Lectures in Physics. As always, it's an absolute pleasure to see so many people in the room. And this year as well, we're live streaming as well, so hello to those of you online. My name's Susie Shee. I'm an Associate Professor here at the School of Physics at the University of Melbourne. And for about the last 6 years, I've been the curator of this wonderful winter series of the July Lectures in Physics. How many of you were here last week? Oh, we love you so much. Thank you for coming back. We have such a dedicated audience. It's quite a phenomenon to see 300 or 400 people come out on a Friday night in winter to hear all about physics. As those of you were here last time will know, this year's theme started last week with a lecture about Pacific sky knowledge, and now this week and for the rest of the series, we move into some really exciting lectures about quantum science because it's the International Year of Quantum Science and Technology. Now, before we get going this evening, I'd just like to acknowledge the traditional owners of the land on which we live and work and hold these lectures, which is the Wurundjeri Woiwurrung people of the Kulin Nation, and to acknowledge any Indigenous First Nations and Torres Strait Islander people here tonight who might be in the room. So you're very welcome, and these lands were never ceded. So welcome everyone. I actually have an extra special little announcement or mini lecture for you tonight, which is a bit exciting. Did anybody pick up a small item from a bowl on a table on the way in? Okay, well, all is going to be explained because before I introduce the main speaker for tonight, it is my absolute pleasure to introduce Hunter Johnson. Hunter is a student here, a second-year PhD student working in the research group of David Simpson, who will be our main speaker for this evening. She did her undergraduate, master's, and now PhD here at Melbourne. And to find out what she's working on, I'm going to hand over to Hunter Johnson. Welcome to the stage. Thank you.
Hello, are we on? Excellent. Thank you, Susie, for the introduction, and thank you all for coming out. We love to hear about physics on a Friday night. Very good. Here we are. This is the crux. This is a working title, but a title for the thesis nonetheless. So we're going to break this down for the duration of this quick little warm-up round. Recording intracellular action potentials with colour centres in diamond. Cool. And we did have a bunch of we did have a bowl. Can we get some crystals up in the air? Oh, fantastic. We love that. Love to see it. Okay. So to break down first, we're going to start at the end, talking about colour centres. What is a colour centre? So let's hold these guys up again. You can see through this is a quartz crystal. Quartz usually transparent, but this one has some veins of darkness, of some smoky wisps in it. And those wisps represent aluminium defects or deposits that make their way into it while these crystals are growing. Now, this aluminium changes the optical properties of this crystal, and that's what gives smoky quartz its colour. It's what gives a lot of crystals their colour. And that's something that we're really interested in this group. And I want you to think about this particular optical property here. Imagine if you could change it. You could make the crystal clear, and then you could turn the smoke back on. And you could turn it off, and you could turn it on again. That's kind of what we do in our group. But this is this is quartz. We're interested in diamond in particular. Cool. So this is a diamond, not to scale. The ones that we work with are a lot smaller. Usually 4 mil by 4 mil, 2 mil by 2 mil is what I'm interested in. And if we zoomed right in there, we would see this nitrogen vacancy center. Kind of like the aluminium in the quartz, it's a color center. When you pump a bunch of green light into it, you get red light back out, so it's fluorescent. And that fluorescence changes in response to the world around it. In particular, magnetic fields, electric fields or voltages, and temperature. They all modulate this fluorescence, which we can record with a camera. Our group are getting pretty savvy at making them, at controlling them, at sensitizing them, and that's sort of a lot of what we do in the Simpson group. Okay. Part 1 done. Next, action potentials. What's an action potential? So we're made of things like this. This is a neuron or a cartoon of a neuron. It's kind of like a current carrying wire in a certain way. We have inputs that come in, we have inputs that come in on this side, and then current flows along this axon here, and then outputs and communicates with the cell down the stream. This is the particular kind of cell that I'm really interested in for my research, so I'm detecting voltages associated with this. We are made of a bunch of these cells. They are constantly zapping, they are constantly talking to each other. When you want to grab a glass of water, when you're having a thought, they're just constantly zapping and zapping and zapping. So that electrical signature is the signal of interest for my research, my PhD. And on this note, I'm really standing on the shoulders of my supervisors, David, Nikolai Donchuk, Dan McCloskey, all have done a lot of work over the last few years that I can just kind of jump on top of in a certain sense. So thank you so much to you guys as well. All right, we're getting our way through. Recording, intracellular, absolutely. So I said these cells are electrically active. If we have a sensor that's on the interior of the cell, that electrical signature that I'm interested in is a lot stronger. So we want to get inside. Now, I made one of these guys. I've made a few thousand of these, I think now, using a process called nanofabrication. This is about, you can see the scale bar here, is about 400 nanometers. Tiny, tiny, tiny things. We want to put fluorescent sensors, our fluorescent defects, the nitrogen vacancy center, in the tip. They're going to flash on and off. This is the pitch. Anyway, we're getting there. We're getting there. And this is the goal. And that's sort of the crux of my sort of PhD is getting fluorescent sensors in the tips of these diamond nanopillars and recording from electrically active cells. Cool. Good. Great. Okay. Yeah. To say that again, I suppose, we have, again, not to scale, a diamond. We're going to put a bunch of these guys on it, and then we're going to put a bunch of cells on it. And that is sort of, I guess this is a timeline of my PhD, I think, here, just all in a few slices. It's a very collaborative project, a lot of collaboration through the Centre of Excellence that you'll hear more about from David, Cubic, collaborating with neuroscientists and people who can do cell stuff good. I do more of the nanofab stuff good, but yeah. Thank you very much, and enjoy the rest of the evening as well.
Thank you. Thank you so much. That was absolutely incredible to be able to describe something so complex in 5 minutes. It's actually much harder than the job that our main speaker has to describe his stuff in 45 minutes, as everybody who's ever given a talk of only 5 minutes will know. But I thought I would bring to this audience, some of the real work that's happening in our research labs by our students, not just by our academics. So Hunter will also be available at the reception after, and you can ask questions about Hunter's research in the Q&A at the end as well. That goes for those online. If you'd like to pop a question for Hunter into the Q&A, we can manage those at the end of the session. But to the main event, our main speaker for this evening is Associate Professor David Simpson. David has worked at Melbourne University, I wasn't aware of this, since 2006. Now, did any of you hear Hunter and I on Triple R last Sunday? Is anyone here because of that? A couple of people? You will have heard Shane Huntington, the host of the show, make a joke at the time to say, oh yeah, I hired David to the university, and it's something that he was very, very proud of in the interview about the research that Hunter's doing in David's group. Well, you can go back and listen back for those of you who weren't there. But David has now gotten to a point where he has quite an enormous group working, by my standard, working in sort of biophysics and the quantum aspects of that. And so without further introduction, because I know he has a lot to get through and some very exciting demos to show you, I'd like to welcome to the stage Associate Professor David Simpson. Thank you.
Thank you very much, Susie. Can people hear me okay? Excellent. Thank you very much for coming out on a cold Friday evening. I really appreciate it. It's fantastic to see such a large in-person audience, and thanks very much for those that are tuning in online. It's a real testament to the success of these July lectures that we have, and in particular, I want to thank Susie for her curation of the lecture series this year. which is about quantum technologies, which is a particular passion of mine. So thanks for all the hard work behind the scenes, Susie. I also want to particularly thank Hunter for that wonderful introduction. That really sets the scene for what I want to talk about tonight in terms of engineering, fabricating these new types of sensors made from colour centres in diamond, and then deploying these sensors into interesting problems in biology and help us to understand biological systems in new ways. So that's what I hope to convey to you tonight. So the title of my presentation really focuses on quantum sensing. So I want to talk to you about what are quantum sensors, how can we exploit quantum sensors to do interesting things, and how do we engineer them? What sort of materials do we make these sensors out of? And tonight I'm going to focus our attention on diamond materials in particular, because that's a key focus of our group. And I want to talk about how we can apply these diamond materials to answer interesting questions, as Hunter's just alluded to develop new platform technologies to image the electrical signals from complex neuronal networks. And I'll talk about how that works and some of the real breakthroughs that we've had in that area of late. I also want to talk about some of the work that we're doing at the very small scale end, looking at fundamental how iron is loaded into one of the most important proteins in our body, which is ferritin. And so I'll tell you more about that in the second half of the presentation. And then I've got a very special experimental demonstration that our group's been working on for a number of weeks, which hopefully blows you all away. So that's the plan for this evening. Now, to get started, I just wanted to reference back to this. It's a really important year for us quantum physicists. So it's the 100th anniversary since the discovery of quantum mechanics. And so UNESCO announced last year that 2025 was going to be the International Year of Quantum. And for us, that gives us an opportunity to really communicate to the public about the impact that these quantum technologies can have in life and on society going forward. So the real focus for us in these next series of lectures is to communicate to you guys about what's happening in this area. It's also an opportunity for us to establish new collaborations. So this year there's been lots of new opportunities to work with groups not only within Australia but internationally and really get the exposure up in this particular area. And there's a lot of work that's going on in terms of the policy development of quantum technologies. And one key example of that I want to point out was something that happened in 2020. So CSIRO published this roadmap. This was a quantum technology roadmap. And what it did was survey all of the people working in quantum technologies. It surveyed a whole bunch of industries, and it looked at what are the potential impacts of quantum technology out to the future. So we're looking to 2040 in this particular instance. And they broke that down into several different categories. So they had a look at the impact of quantum computing. This one. Quantum computing, quantum sensing, and quantum technologies. And they broke that down into 3 individual markets. And they had a look at how big could these potential markets be, how many new jobs could be created off the back of those new industries. And there were some pretty pleasing numbers to see. Now, that was published in 2020. Since then, these numbers have already been updated. The markets are projected to be much bigger than that. The number of jobs that are created are also expected to be much bigger than that. So this is a rapidly evolving field, and we're finding new applications and new markets all of the time in this area. Now, following that document, the federal government then released a national quantum strategy, and that was published last year in May. And pleasingly, it was great to see that the University of Melbourne had 2 use cases published in that national quantum strategy. And those use cases talked about how we could use quantum sensors to improve medical testing, and how we could use quantum sensors to help inform more accurate navigation systems. So they were 2 examples from the work that we were doing here. This national quantum strategy really outlines how Australia will support this industry into the future and what's required to actually get this industry off the ground, to see that economic benefit and to see these new jobs progress. And so that's being rolled out now. That's also led on to a whole range of other policy documents. So the Army's developed their quantum technology roadmap. The Queensland state government's released a quantum strategy, and quantum technologies are a key part of the AUKUS Pillar 2 agreement that the federal government signed with the US and the UK. So there's a lot of activity in that space that's really trying to support the growth of quantum technologies. And I hope to convince you tonight that it is worth supporting these efforts in terms of their near-term potential, particularly around quantum sensing, which will be the focus for tonight's talk. Now, in this National Quantum Strategy, they did an excellent job of surveying all of the different technologies that are being worked on, all of the different quantum materials that are being explored in all of the different states. And you can see that there's a lot of activity going on all around Australia in this area. And what I want to do tonight is tell you about what's happening here in Victoria, and my particular focus is going to be around these materials, these quantum materials, which revolve around defect centres in diamond. And this work has recently led to the establishment of a new Centre of Excellence, and this Centre of Excellence is focused on quantum biotechnology. And so this Centre of Excellence was launched last year, and the mission of this centre is to develop new quantum tools that can be applied to the life sciences to understand how biological systems work and function at unprecedented levels and speeds. That's what we aim to do. And so the University of Melbourne is the second biggest node of this Centre of Excellence. It's a $35 million Australian Research Council funded activity. It involves 5 other universities around Australia, and we're all working towards that goal of having impact in the life sciences. Now, in terms of that centre, we've got 3 main themes that we're looking at, and some of these themes overlap with what I want to talk about tonight. So the themes that we're working on in that centre evolve around understanding molecules and how molecules work, change, evolve over time, and how do we measure those. We then want to understand how does that molecular activity lead to emergent behaviour in cells. And then once we can understand this emergent behaviour, how does that then lead to more complex things like neurons being activated in our brain or in a dish. And so we're going from the very small scale to the very large scale. And to do that, we can apply a whole range of different quantum tools. And tonight I'm going to tell you about how we apply these defect centres in diamond to address this neurological challenge, which I'll talk a little bit about, and also how we use it to study this very important protein, ferritin. So that's where we're going. So first I want to touch on the brain. And the real motivation for us looking to understand the brain in better ways is that neurological disorders have a profound impact on our society. And so at the moment, this is estimated to be a $16 trillion problem, not to mention all of the people, families that it impacts. And one of the problems is it's very difficult to find drugs that successfully treat neurological disorders. And you'd be surprised to know that at the moment, given the scale of that problem, there are only 360 drug trials for a neurological drug running at the moment. It's only 360, when this is such an enormous burden on our society. And so the reason that number is so small is because 94% of those drugs that they're trialling right now will fail. They won't be effective, or either they won't be safe to put into a human. There are a whole range of different reasons why these drugs fail, but we haven't got the appropriate tools at the moment to understand how these drugs are acting on these neuronal networks. Now, this is a big problem for pharma because it costs pharma up to $2 billion a year pursuing these drugs that are failing, and it's taking up to 8 years to fail these drugs. So this is an enormous cost burden, and it's an enormous timeline that's being wasted because we haven't got the right tools we need to understand how these drugs are acting on these complex neuronal networks. And that's something that we want to try and develop new technology to help assist that process. And that's what I want to talk to you tonight about. And so there's a lot of work going on in this area to understand how drugs impact neurological disorders and diseases in general. And I like to categorise them into 2 different areas. So there's a lot of work happening in what I would call systems biology. So this is looking at how do the cell's genes, proteomics, metabolomics change from a healthy system to a diseased system. So what's happening with the inside the cell that's changing the makeup? So there's a lot of work going on in this systems biology area, and there's a lot of good tools that we already have to do a lot of that stuff. The second question is those modifications that happen at a cell level, how does that manifest in terms of the excitability of the network? So how does that affect the firing dynamics of a neuronal network, the function of a neuronal network, how these neurons communicate with one another? We have some tools in that area, so we can do functional MRI, for example. We could do magnetoencephalography, where we put a helmet on and we can measure the magnetic signals from the brain. We could do EEG, where we measure the electrical signals from the brain. That's all at the whole head kind of level. And so we've got the large-scale stuff covered. What we haven't got covered is what happens at the individual neuron level. What happens to these individual connections that happen to these neurons? How do we image these? And once we can image them, then we can understand how they fundamentally connect together. What are these connection rules? And then how is the excitability of that network changed in the presence of a drug, for example? And the challenge here is that the signals we're trying to measure are very fast. So these action potentials that Hunter talked about in the beginning happen over 1 millisecond. So they're very quick. And they're happening over a spatial scale of about 1 micron, a millionth of a meter. So you need a very sensitive electrode to be able to detect these action potentials in the first place, and it needs to be tiny if you want to get the spatial resolution that you need. And so the tools that we really have to study this complex beast, the way that we do it, the way biologists begin to break it down, is we culture the neurons in a dish, and we can do that very well. These neurons form these networks, these complicated networks, and we can take these beautiful images that tell us this neuron's connected to this one, etc. But it's a static image. It doesn't tell us how the electricity is flowing in that network. It doesn't tell us which neuron is talking to which. We don't know anything about its connectome. And that's something we'd love to change. And so Hunter mentioned a little bit about these action potentials and what they are. It's these neurons firing electricity and propagating voltage. Now, you can measure this in different ways. You could get an electrode and stick it in to the neuron, and that was awarded a Nobel Prize back in the 80s. And so you can get what's called a patch clamp. You can put this electrode into the cell, and what you will measure is something called an intracellular voltage. And that voltage is pretty large. It's 100 millivolts. But as I said, it happens very quickly, over 1 millisecond. So that's one way you could do it. And when you patch into a cell, you can only typically record from that cell for an hour or two, and then it will stop firing. Okay, so there's limited information that you can get from that. The other way to do it, is to position the electrode very close to the membrane of the cell, and you will measure what's called extracellular potential. And that's the electric field that propagates on the outside of that membrane. That looks a little bit different to the intracellular signal, but the important part is that that's a much weaker signal. So when we're outside of the cell, that's only about 5 millivolts. Okay, and again, it's over 1 millisecond, so it's a very difficult thing to measure. Nevertheless, there's been tremendous progress made, and here are some of the state-of-the-art devices for measuring that electrical activity. These are called multi-electrode arrays. And no surprises, it's an array of electrodes that record these voltage signals. So you can see here, this is a CMOS chip, and you can see these little gold pads here. Each one of these gold pads is recording the voltage of neurons that get cultured on top of that chip. Okay, so fantastic device. They're measuring the electrical extracellular activity of these neurons. The issue with these electrical approaches is we've got these electrode pads, and we can only bring these pads so close together. If we bring them too close, what happens is you get electrical crosstalk between the pads, and it no longer is representing the signal on top of that pad. That's one of the problems. Another problem is that these electrical pads require an amplifier underneath to amplify that very weak signal before sending it to your signal processing unit. That also takes up precious area. So there becomes a point where the density of these electrodes hit a limit, and there's a technological limit that's been hit at the moment. So the number of electrodes that I can jam in to a millimetre squared has been stuck at the same density for the last 10 years. So this electrical approach has really hit this technological limit in terms of density. Even so, we can still do recordings from it, and you can start to see the problem here. So this is a neuronal network cultured on top of one of these multi-electrode arrays. This is work done at Harvard. So this was beautiful work, but what you can see is each one of these square pads here is one of those multi-electrode arrays, and it's recording the voltage across that whole pad. And you can see that there's much more complicated structure on each of these single pads that the MEA just can't detect. It just detects the average from that. So we can get some inferences about what's happening to the firing dynamics from this network in general, but it doesn't tell us how these neurons are connected, and we can't do any optical imaging to work out the routes of and the connection rules of this network, which is something we really want to get to. So this electrical technology is fantastic for long-term recording because we're looking at the extracellular potential. So that's great. We can do that at high speed. The problem is it significantly undersamples that network. It doesn't look at every individual neuron and how the electrical signals are propagating across those. So that's the state of play for electrical devices. We could move into the optical domain. And in 2010, science announced optogenetics as the breakthrough of the decade. And optogenetics is a really remarkable thing. So in optogenetics, what you can do is you can transfect your cell with a virus, and that virus will cause the cell to produce these channelrhodopsins. And these channelrhodopsins, what they do is when you shine light onto that channelrhodopsin, that creates a voltage, and that voltage can stimulate a neuron. So now you've got an interface where you can shine a light on a neuron, and you can cause that neuron to fire. So you could start to then probe, you could start to stimulate anywhere you like in that neuronal network, and it will then fire off an action potential. Then what they did was combine that with a voltage-sensitive dye, which is also transfected into the cell, that could read out the voltage. So there are dyes that respond to a voltage. So in the presence of a voltage, that dye will start to glow, and it will tell you that there's a change in voltage at this particular point. And with this optogenetics, we can now, or not we, but the team at Harvard were able to produce this beautiful image, which really demonstrates where we want to go. This is a single cell. You can see the axons are spreading out from that single cell, and that cell is repeatedly excited by this blue light, and that excitation causes a series of action potentials that then propagate out into the network. Okay, so this is a beautiful image of what we would love to be able to do. Now, this was really groundbreaking work that happened only a couple of years ago. The issue is, at the moment with these voltage-sensitive dyes, you need to do this experiment 100 times and then average all of those images to get something like this, to get a movie like this. Okay, which is great. So basically what's happened is that they're sitting on their cell, they excite it, they record, they excite it, record, excite, record. Let's do that 100 times, and then add up all of the images, and then you get this beautiful movie. What we would love is if we could watch that in real time, where we don't have to stimulate it, where we can just watch what the electrical flow is of that neuronal network in real time and create these high definition movies. That's where we'd love to go, and that's what our group is really focused on doing. These optical approaches have been wonderful in terms of improving our spatial resolution. So now we can see individual axons and individual cells firing. So we've solved the problem that the MEAs had. But the issue is that these dyes, they bleach over time. So the amount of light that they give off over time starts to decay. And that means we can only typically record from these guys for maybe 5 minutes or so. So we don't have that long-term recording capability that we get with the MEAs. So what we'd love is to have a combination of the properties that the MEAs give, extracellular potential, long-term recording, with the spatial resolution that optical approaches give. And that's where we think Diamond can play a real role. And I'll tell you why we think Diamond is a good choice for this. So in the optical approaches, we can get that beautiful spatial resolution that I talked about, but the problem is these optical reporters die over time. And so if you look at these single molecules or fluorescent proteins or voltage-sensitive dyes, they only typically last on the order of minutes. If you look at defect centers in diamonds, similar to what Hunter talked about in the beginning, these nitrogen vacancy defects, if you shine a green laser onto a diamond and you look at the light that comes back from that diamond, that light is perfectly stable over hours, over days, over years. It doesn't change, it doesn't bleach or blink. And so if you've got a defect that gives you this beautifully stable intensity, the question is, could we use this intensity? Could that intensity in some way be modulated by these electrical signals from the neurons? If they could, that would be a fantastic sensor. We'd have the resolution we need. We wouldn't have to transfect the cell lines, and we could get a direct readout of this extracellular potential. So we started to look at these defects in more detail several years ago for this particular purpose. Now, the University of Melbourne has a long history in diamond research. So we've been growing diamond for over 30 years here. And here is one of the first diamond reactors that was installed at the University of Melbourne. This was a recent upgrade in 2010. And then more recently, we've got this beautiful machine. This machine produces the most pure diamonds in Australia and can be used to grow quantum-grade diamond material, is what we call it. And so as the purity of this material has improved since the 50s, when it was first engineered, so too has the number of applications that we can apply it to. And now there's a whole range of quantum technologies that are enabled by the purity of this material, which is very exciting to see. Now, Hunter talked about the fact that if you can synthesize a diamond, if you can synthetically engineer a diamond and not dig it out of the ground, you can start to put whatever you like in that diamond. You can put different atoms in that diamond. And when you do, as Hunter said, you change the optical properties. You change the color of diamond. If you put lots of nitrogen into a diamond, you'll make it beautifully yellow, like this. If you put lots of boron in a diamond, you'll have a beautifully blue diamond. And so this is used in the gem industry to grow all of these beautiful colors. But we want to use it for a scientific purpose. So now that we can engineer these defects where we want, at whatever thickness we want, we can start to unlock a whole range of new applications. Now, talking about this NV center specifically, it comes in 3 different charge states. So there's a handle we can play with in terms of the charge of this defect. It can either be negatively charged, it can be neutrally charged, or it can be positively charged. And this charge degree of freedom allows us to we can read out the charge degree of freedom just by looking at the light that comes back from these defects. So if I shine a green laser, which is what's happening here, onto a diamond that's rich in negatively charged NVs, I'll get a beautiful red fluorescent. The diamond will glow this beautiful reddish pink. If my diamond, on the other hand, has lots of nitrogen vacancy centers that are in the neutral charge state, I get a beautiful orange color. And if they're in the positive charge state, I don't get anything at all. It's completely dark. And so we've got this situation where we could modify the charge of these defects, and if we do that, it changes the color of the light that's being emitted by that diamond. And it does so very quickly. It can do so on a timescale of microseconds to nanoseconds. So it's fast enough, it can switch color fast enough to be able to respond to something like the electrical activity of a neuron. The challenge for us was, how do we turn this color change into something that would be sensitive to these action potentials that are emitted by a neuron? And that's something that we've been working on over the past 7 to 8 years. And the way that or the place where we've landed at the moment is we started to put these defects into diamond. So as Hunter mentioned, so here is a diamond chip. We implant nitrogen into that diamond to create this beautiful 2-dimensional array of these NV defects in a diamond. And then we wanted to understand, what can we change about the surface of the diamond to see if we could change or modify these charge states in some way. And it turns out that if you oxygen terminate the surface of a diamond, you get this beautiful pink glowing red fluorescence from the negatively charged NV centers. If instead you do not oxygen terminate, instead you hydrogen terminate, you turn all of those NVs into the positive charge state and you don't get any fluorescence at all. And then you can imagine if you have a mixture of an oxygen-terminated surface and a hydrogen-terminated surface, then we get this neutral charge state, and the majority of the NVs are in this neutral charge state, emitting this lovely orange fluorescence. So here, what we're changing is the surface termination. That's changing the local charge environment that the NVs experience, and based on that charge environment, they can switch colours, which is exactly what we need to be able to detect these neurons. And so this was the first demonstration that Dan and Nikolai performed several years ago now. So this was part of Dan's PhD. So Dan is sitting somewhere over there. So you can talk to Dan at the end of this event. And Nikolai was the postdoc working on this at the time. And this was a beautiful demonstration. So here what we did, we took our diamond with those near-surface NVs, we put it in a conductive solution, and then we had a tiny micro pipette. And that micro pipette could inject charge into that solution. And so the idea here is that if the pipette injects a positive charge to the surface of the diamond, that's going to modify the population of the NVs to be in the neutral state, and we're going to get lots of orange fluorescence. Whereas if the surface of the diamond is negatively charged, it's going to put the diamonds into the positive charge state, and we'll get no fluorescence. So there'll be areas where the diamond's dark, and where there's positive charge, we'll see fluorescence. And that's exactly what we saw. So when you apply a voltage, here a voltage is of order 100 millivolts, we see this dramatic increase in fluorescence from our chip, which says that it's responding to the charge that's flowing onto that surface, and as the charge diffuses away, our fluorescence decreases as well. So we're getting direct readout of the surface charge produced by this little micro electrode here. Okay, so this was a great start. Since then, we've spent the last 2 years working with colleagues at the Florey Institute for Neuroscience and also other colleagues here at the University of Melbourne to understand how do we culture neurons onto these chips to do these recordings. And in particular, we've been working a lot with Jenny Gunnarson's group, and Catherine Munro's done a lot of the optimization of these cultures. And we're now at the point where we can culture these neurons onto our sensors. And as of about maybe a month ago, I think, Dan, or a couple of weeks ago, which was very timely for this lecture, we recorded the first ever action potential measurement in real time, single shot, using this technology. So here is an image of a single cell, a single neuron. It fires an action potential, and we're measuring the extracellular activity of that action potential. So you can see that this image suddenly fires and then disappears. That's the action potential. And we can image that in every pixel. And so this image actually represents the current record for the highest ever recorded extracellular potential from a single neuron. And so we're doing that in real time. Okay, we don't have to do the 100 averages that I talked about before. So now we can start to watch how the electricity flows and how these voltages flow in a neuronal network. And so what we want to do, you can see that this is very small scale here. So here we're just looking at a single cell. So now we're redesigning our microscope so that we can image this whole entire network and then see where this information propagates out. And we can start to watch how those neurons are talking to one another. So this has been a tremendous advance for the group. We're very excited about where we can take this, and now we're working with lots of neuroscientists who are interested in applying this sort of technology to their research. And as part of that, there's also been a startup company that's been established, which is called Chromos Laboratories, and Nick, Dan, and Eliza are really leading that effort. And the focus of Chromos is to really develop this as a tool and as a platform that neuroscientists can use, and hopefully we can go back and start to culture networks onto these chips, have a look at how drugs are affecting individual neurons in this complex network and providing information that you just didn't have access to before. And if we can change the game in terms of moving that failure rate, even from 94% to 90%, that'll have a dramatic difference in terms of development of neurological drugs for these terrible disorders. And we're doing that in partnership with a number of different partners, some pharmaceutical companies here in Melbourne and also the NIH over in the US. So that's the story about neurons and what we're doing in the brain theme of this Centre of Excellence. In the second half of this talk, I want to switch gears a little bit and have a drink, and I'll talk about this important protein, ferritin. So please have a look at this structure and I'll tell you a little bit more about that. So I started off at the big scale, looking at networks of neurons. I now want to go into this small scale, and I want to show you some of the work we've been doing, understanding how iron is loaded into these important proteins. And I just want to give you a picture about how small a ferritin protein is. Okay, so in this slide here, here is a single strand human hair. So that's about 100 micron in size. And then if we go down in scale, so this is like a fine beach sand, a single grain of salt. We keep going down. Here's a annoying dust particle at 10 microns. So that's 10 millionth of a metre. We can go down to a red blood cell, is around 7 millionth of a metre. Bacteria is around 1 micron, very small. And then we get into the viruses. So here's the nasty coronavirus. It's about 100 nanometres in size. You keep going down. We're into Zika virus, is about 45 nanometres in size. Go smaller again, you're down to 12 nanometres. That's the size of a single ferritin protein. And this protein looks like this. This is a 3D print of the actual crystal structure of a ferritin protein. So it looks like a spherical ball. And the role of this ball is to trap and store iron. Okay, and I'll talk about how we measure our iron status in our body in a second. But I just want you to picture this as a spherical ball that's capable of loading iron into it. Okay, that's where we're going to start. Now, iron is incredibly important within our body, and it's managed via a number of different pathways. If we have too little amount of iron in our body, we become iron deficient. If we let that go, we become iron deficient anemic. Okay, and the path back from iron deficiency anemia is very long. If you're iron deficient, often you can take iron supplements and you'll be fine. The challenge is it's very hard to identify when someone is iron deficient. And I'll tell you a little bit more why that's the case. On the other hand, you could be iron overloaded. So one of the most common genetic disorders is something called hemochromatosis. It's when you have too much iron in your body. And the way that we deal with too much iron is very simple. We just bleed the patients regularly to get the iron out of your body. It's that simple. It's not a great or effective treatment, but it works. That's the best we can do at the moment. So if you've got too little iron, it's a problem. If you've got too much iron, it's also a problem. And I want to talk about the techniques, the ways in which we measure our iron status at the moment. So the World Health Organization says that ferritin, this little guy, is the best biomarker we have for measuring our iron status. The most common way we measure that is through a simple blood test. You go to the doctor, you fill out your form, you go and get a blood draw taken. That blood draw goes to a pathology lab. They take the serum from your blood, and then they measure how much ferritin is in that serum. Okay, and the number that you get back tells you the concentration of these particles in a certain amount of serum. Okay, what it doesn't tell you, that test, is whether the ferritin is empty, whether there's no iron in that ferritin at all, or whether it's full of iron. So this ferritin protein can have no iron atoms in it at all, or it can have up to 3,600 iron atoms stored in it. Okay, but the tests that we do at the moment do not test for what's inside this protein. All it simply does is ask the question, how many of these have you got? And if you've got any underlying health condition, if you've got any chronic condition, often your body produces more ferritin, and that ferritin is often not loaded. Okay, so that can give you an elevated ferritin measurement when you might actually be iron deficient. And so that's a problem that we want to solve. So the current blood tests are cheap, but they don't work for half of the population that's living with a chronic condition. And this also disproportionately affects women. So this is a really big problem for women. So it correlates, the iron status correlates very well in healthy people, which is great, but they're not the people that are going to the doctor to get their regular blood test, okay? So we want to do better than that. There's an alternative. You could go for a really accurate method, but it's invasive. You could sign up for a bone marrow biopsy to measure your iron levels, but there's not too many people signing up for that. Okay, to get a big needle in your back, extract the bone marrow, and then basically you put that on a microscope slide, you image where the iron is in those proteins, you count them up, and you get it direct. So it's a direct measurement, but it's rarely used. There's another method to evaluate your iron status, which is through magnetic resonance imaging, MRI. And this is another good method because it's a direct detection of the iron that's present in your body. So when you go and evaluate your iron status through an MRI, you do the MRI scan on your liver. Your liver is the area that contains the most amount of iron. And what the MRI is doing is measuring the influence of iron on the water molecules in your body. And I want to talk a little bit about the physics of how that works in an MRI. And then I want to tell you about how we could do a similar thing with these defects in diamond, but do it in a much more sensitive way. So how does an MRI work? What are the fundamental principles of an MRI? You go inside this big tube of an MRI. What does the big tube do? What's the role of that tube? Produces a magnetic field, all right? And it produces a really big magnetic field. The magnetic field produced inside the center of an MRI machine is somewhere between 1 Tesla to 7 Tesla, okay? That's like 6 orders of magnitude or 5 orders of magnitude bigger than the Earth's magnetic field. It's enormous, all right? And we need that magnetic field for a very important purpose. Now, our bodies are predominantly made of water, so we've got lots of water molecules floating around. So water is 2 hydrogen atoms and an oxygen atom. And for the purpose of this talk, the way I want you to think about these protons, these hydrogens, is basically acting like a little bar magnet. Okay, they've got a north and a south pole, and that bar magnet can exist in some direction, which I'm defining with this arrow here. Okay, what really goes on, this nuclei has a fundamental property called a spin. Okay, I'm not going to go into too much detail about that, but just think about this as a little bar magnet pointing in that direction. Now, when we walk around and when you're sitting in your chairs now, you've got lots of these water molecules, they're all moving around, and you've got all of these bar magnets pointing in different directions. So if you were to add up the magnetic field from all of those little bar magnets, the total field that would be produced would be approximately 0. Okay? If it wasn't, we'd be attracted to each other magnetically, which we're not. As much as I love you, I'm not attaching tonight. So this is what happens in the absence of a magnetic field. What happens when you apply a really strong magnetic field? What you do is you align all of those bar magnets in the direction of the applied field. So if I turn the field on, or if I go inside the MRI machine, that magnetic field aligns all of those proton spins in your body, okay? The majority of them align with the magnetic field, and there's some that are anti-aligned with that field. Now, what we want to do is we want to understand how these protons or these spins relax. How do they decay in the presence of their environment? And I'll tell you about why we want to do that in a second. But the next thing we do in an MRI is we apply a radio frequency pulse. Okay, so once we've aligned all of our protons like this, we apply a radio frequency pulse at a specific frequency that tips all of those protons by 90 degrees. Okay, so we rotate their magnetization so that they now point at 90 degrees. Now, the protons don't like existing in this state here. They would rather align with the large magnetic field. So what happens is once we flip them, they then relax and realign with that large magnetic field. Okay, as that happens, they release energy in the form of an electromagnetic wave at a specific frequency, and we can detect that radio frequency signal using a pickup coil. And that pickup coil is on the inside of that MRI machine. Okay, now importantly, that RF signal falls off as 1 over r cubed. Okay, and that's a bad scaling if you want to detect a signal, if something's falling off as 1 over r cubed. And if your coils are a long way away from your body, that signal, that magnetic signal that you're trying to pick up from these water molecules decays. And that's one of the reasons why MRI is not very sensitive. Okay, because these coils are a long way away from the water molecules that are producing the signal. Sometimes when you go in an MRI, they might put coils closer to your chest or closer to your head. The reason they're doing that is because you're closer to the magnetic field, your signal will increase, the sensitivity of that scan will improve. Okay, you might get a better resolution scan in that particular case. But the measurement that we want to do in an MRI to infer our iron levels looks something like this. So as I said, we tip all of our protons by 90 degrees, and then we watch how long does it take for them to relax back and align with the magnetic field. And that time has a characteristic time scale associated with it, which is called the T1 spin relaxation time. It's how long does it take for the spins to relax back, okay? Now that time, that spin relaxation time is dependent on all of the other things that are surrounding these water molecules. So if you've got ferritin, if you've got lots of iron nearby these water molecules, that iron has unpaired electron spins. And electron spins have a magnetic moment that's 1,000 times stronger than a nuclear spin, than these proton spins that I'm talking about. So now, if you've got any kind of iron hanging around, you've got a huge bar magnet now that's moving around and fluctuating, and that's causing lots of magnetic noise. And what that does to the proton spins is it causes those proton spins to relax faster. Okay, so we tip them. If there's iron around, now they relax really quickly due to all the magnetic noise surrounding that. And that's exactly what we measure in an MRI. Okay, we're just looking at this relaxation rate, and that's how we build up an image. And so if you've got hemochromatosis, for example, which is a common disorder that's diagnosed via MRI, one of the key indicators is you build up iron deposits in your liver, which look like this. And in an MRI, you might have a scan that looks like this. And here the dark patches represent regions where you've got a short T1 time. That's the region where you've got lots of iron that's causing the water molecules to relax faster. And then the white regions in this scanner where you've got a long T1 relaxation time. Okay, so there you haven't got a huge amount or a huge accumulation of iron. So this is a beautiful technique. We can directly say where the iron is. We can quantify the amount of iron that's present in your liver. Fantastic for iron overload. So we can do, and we can diagnose and we can treat using MRI for iron overload. Unfortunately, these scans are expensive. They're over $600 a pop. So in terms of doing management, you don't want to go for a $600 scan to understand is now a good time to bleed or not for iron overload. Okay, so that's prohibitive. The other thing that is prohibitive about this technique is that it's only working for iron overload. It's not sensitive enough to get into this iron deficiency regime. And so we'd love to be able to push into that space. We'd love to use this principle of magnetic resonance imaging and apply that to diagnose people that are iron deficient. So, how can we do that? Well, this story dates back to 2009. So here was a proposal put forward by Jared Cole and Lloyd Hollenberg. And this was a theoretical proposal at the time that said if you had a quantum sensor that was nanoscale in size, so very small, and you position that quantum sensor very close to a ferritin protein, for example, that's got iron in it, then the magnetic field from that iron should disturb your quantum sensor if you can get it close enough. And as I showed you before with the neurons, we can position these NV centers in diamond within 7 nanometers from the surface. So we can get these quantum sensors super close to a surface, And if we put the ferritin on that surface, now we should be affected by the magnetic field that's coming from the iron inside that ferritin. So this was a beautiful theoretical proposal which said if you could come up with a system to do that, then it's possible, and they even modelled how the signal would look for a ferritin protein, looking at the change in the quantum properties of a sensor. And so this has taken us 15 years since that proposal to actually get to the point where we can now demonstrate this idea. So I don't want to say that this was a quick demonstration, but we've put a lot of effort into understanding how to do this with NV centers in diamond, and now we're on the cusp of some really exciting results. The other thing I like about this slide is I've got a photo of Jared and Lloyd in author order. So it was a Cole and Hollenberg paper. So when I saw them out one night, I had to take that photo. It was too good to refuse. But these have been real visionaries in the field of quantum sensing, and Lloyd and Jared also do a lot of work in quantum computing as well. So how can we take these principles of MRI and apply them to the nanoscale with these defect centers in diamond? How do we do that? That is what I want to demonstrate to you tonight. We're not just going to talk about that on a slide. So one of our PhD students has put together a live demonstration to show you this and how we actually do this. The principles are very simple. So if we have this nitrogen vacancy defect and we shine green light, like a laser, onto that defect, there's a very important thing that happens for NV centers. What happens when we shine our green laser is that those spins, those NV, the spins that are associated with the NV defect, align perfectly. We can align them all by shining a green laser on the diamond. That's really unique. Remember, in an MRI, you have to go in a tube that's got this enormous magnetic field to be able to rotate these proton spins and get them all to align. In a diamond, you can do it just by shining a green laser on it. And we go, bang, they're all aligned. Okay, so that's a really unique thing that we can do with these defects. Once they're aligned, we play the same tricks as what you do in an MRI machine. You wait, you turn your laser off, you let your spin interact with the environment around it, and over time, that spin will start to relax in the same way that the proton's relaxed. Okay? And the NV spin will relax to a mixture of these three spin levels here. Okay, and the way that we read out the spin population for these defects is also optical. We look at the light that comes off these defects. We don't need an RF pickup coil. Okay, so everything gets shrunk down into the nanoscale. We can interrogate it with light to polarize them. We can apply radio frequencies and we can tip the spins if we want to, but we don't need to. And we can read out their population with looking at the red light that comes back. So it's a beautiful system, and what we measure is a decay that looks like this. So it's the inverse of what you would see in an MRI machine. Okay, and when we do that, we were able to show that we can actually measure the changes in this relaxation time as the ferritin gets loaded. Okay, as the amount of iron changes, it should change this spin relaxation rate. How am I going for time? Am I getting bad? Okay. All right, let's do let me do the demo. So what I want you to appreciate here is, let me skip over the amazing results that we did. All right, what I want you to appreciate here is that we can do this now in real time, in a lecture theatre, no massive magnetic field. And I want to show you how we do that with this simple system here. So here, I've got a cuvette, and before I do that, I might introduce the person responsible for this wonderful setup tonight, is Trent Ralph, who's doing a PhD with us. So maybe a round of applause for Trent. I'll get Trent to come up here. So Trent has built a nanoscale MRI machine right here. Okay? So what we've got is a cuvette. We've got a solution containing nanodiamonds with these NV defects. Okay? They're in a solution moving around. If we shine a green laser onto this cuvette, that'll polarize all of the spins into this state that I talked about before. Okay? And if we look at the red light that comes off, when we turn the laser off, we let it evolve. We turn the laser back on, we look at the red light that comes back. We can measure the relaxation rate. And we're doing that from thousands of nanodiamonds that are present in this cuvette. And so here's what the diamonds look like. So they start off very big, they're nice and yellow, they turn black. We make them turn nice and white, and I can tell you about that in the question session if you like. Tad, maybe we can go to the computer and let's see whether we can look at a live demonstration of this. So here we have a cuvette. So you can see at the front here, we've got something green that's glowing. That's the green laser that's going into that cuvette where we've got all of the nanodiamonds. That green laser is pulsing. So if you turn the lights down, you'll actually see that green laser flicker. So you can see that there. Okay, so that's the laser pulsing on and off, on and off. And then the time gets longer between the pulses. And then if we look at the output of what we're measuring on that screen, this is the spin relaxation time that we're measuring from many thousands of nanodiamonds in that solution. Okay, and that tells us about the intrinsic relaxation time of the nanodiamonds in that environment. Now, if Trent was to add ferritin, these proteins that we want to detect, we should see a change in that spin relaxation time from the NVs. Okay, so Trent's going to do what good scientists always do. He doesn't always dress like this to do pipetting. But Trent is going to pipette a solution of ferritin, okay, so these are the proteins that we want to try and measure, into the cuvette that's containing those nanodiamonds, okay? And what we want to see is whether that modifies the spin relaxation rate. And so Trent's doing a little bit of mixing. So what we want now is these ferritin proteins to be moving around in solution. They're then going to come and stick onto the nanodiamonds, and they're going to present a fluctuating magnetic field, which is going to impact this spin relaxation time. Okay, and if we can measure the change in that spin relaxation time, that tells us how much iron, actual iron, is in that solution. That's a direct detection of the magnetic field from iron. So if we go back now to the measurement screen. Okay, all right. You can do your little dance. Okay, so this was the first, that black line there was the first decay that we measured just from the nanodiamonds themselves. And now what we want to do is build up that same image. You have to wait because the network's bad here in the basement of the School of Physics building. Shout out to IT for that. All right, so what we want to have a look at now, you can see the red curve is the one with the ferritin. Okay, so now this spin relaxation time from these same nanodiamonds that we're measuring is modified. And that change in relaxation time tells us that there's iron inside that ferritin, which is the key piece of information that we're trying to get to. Is that ferritin loaded or is it not? And so that's the first time that this sort of technology has ever been taken out of the lab. And so it's a huge credit to Trent and the team here at the School of Physics. So a round of applause for Trent on that. And there's nothing like a deadline to get things done. So this has really helped speed up the progress of the lab to get this demonstrated tonight. I did a lecture like this a few years ago where we had a magnetic sensor detecting DC fields. This year we put that sensor on a plane and we're looking to commercialise that. I'd love to see in 5 years' time us commercialise this and get this into a pathology lab. So hopefully that's where it can go. All right, I might leave it there so that we've got plenty of time for questions. Thanks very much for your patience. Well done to Trent, and look forward to questions.
Thank you, David. And apologies to everybody for having to rush because of our timescale this evening. Hopefully David and Hunter will join us at the reception after so that if there's extra questions, we can ask them there. My first question comes from online. It's actually for Hunter, from Kate, who's watching online. Kate just asks, for your research, if you could summarise the uses of this technology.
Yeah, for sure. So I think David touched on it in the sort of first half of his talk, which is really looking at improving our ability to detect these quite fast, quite small, quite weak signals coming from neurons in the brain. I think there's a lot of really interesting questions just from fundamental neuroscience in actually being able to know how do cells talk to each other on those sorts of scales. But in terms of drug discovery is a really important part. Understanding, like, there's a lot about brains that we still don't know. One of those features is how they connect, how they grow, how they develop, and how they're affected by drugs. So that's definitely the use case there that I'm interested in, I think.
Thank you. Yeah, and that connects the two of your talks very nicely. Great. I'm going to take the next question in the room. We have a couple of microphones in the room, so if anyone has a question, please raise your hand and we'll bring you a microphone. If everyone's still thinking, yep, there's one over the side on the left here. Thanks, Steve. Running the microphone. Thank you.
Thanks. Thank you. Very interesting talk. When you put the diamond things in solution, does the big grey ball there actually stick to the diamond, or are they just in solution next to each other moving around?
Yep. So what we've seen is that they're very attracted to the diamond surface. So they do stick to the diamond surface. It's been very hard to get them off, is what we've found. So it's good in what we want to try and demonstrate here. We want to engineer that interaction to be a lot more specific. So if we want to convert this into a blood test, there are a lot of things that exist inside serum. So it's not just ferritin, there are a whole bunch of other proteins that you've got to worry about. And so our challenge is how do we get the diamond surface to just recognise the ferritin and bind the ferritin so that we can get the change in our signal. And so for that, we're working with a whole bunch of talented chemists to understand how do we engineer that interaction. So for this demonstration, it turns out that the ferritin is slightly positively charged, the nanodiamond surface is very negatively charged, so those two like to come together. Yep.
Great. I'm just going to come back to one that was online from someone called Gemma watching online. Would it be viable to use, I'm not sure who this is for, but either of you take it. Would it be viable to use peripheral neurons for research to reduce the reader density issue? These can be up to 1 metre in length from the spinal cord to an extremity. Not sure if these could be cultured or would have to be sourced from donors, etc. But the question is about peripheral neurons. Who'd like to take that?
Yeah, I can have a go anyway. Yeah, if I you could come in and save me if I needed, maybe. Sure. So for context, peripheral neurons are the types of neurons that connect from our spine and brain, central nervous system. The kind of periphery of our body is peripheral neurons. And they do tend to be quite long. So we're talking, over 1 metre in length. I assume that's the motor neuron that kind of starts up here in your lower back and extends all the way down to your feet. They're very big cells. I believe there is some research on conditions that do target peripheral neurons in particular, like multiple sclerosis. I believe that affects the sort of coating of cells within the periphery. I suppose, if there was a particular sort of target, like a drug target that was specifically for that, I don't see why not. Although I guess you'd need a lot of diamonds to sort of innovate the whole length of it, potentially. I don't know if we have diamonds that big yet.
I think there's a part of the question was about the reader density issue. I think maybe the density of neurons on the diamond structure. Am I interpreting that correctly?
Yeah.
So if you had big long ones, I think the idea is, would that be less dense, and could that help you with the separation out?
Yeah.
That's my
guess
interpretation.
If they're bigger as well, then they would have stronger signals, I suppose, too. Yeah.
Interesting idea.
Mm-hmm. Absolutely. I think it's a horses for courses.
Yeah.
I think not all technologies are good for all applications, and there's a lot of good technologies that would be better at doing peripheral neurons than diamond. I think diamond's advantage is that we have great spatial resolution. It'd be fantastic to look at these individual neurons, but there are, much better ways, and the density problem's not a big deal when you've got a really long neuron.
Yep.
So, I totally agree.
Great. I'm going to come back in the room for the next one. I think there's a microphone on this side. I'll come over this side to this person up here. And then, next, Steve, I'll come to the person that you're going to. After this one. Sorry, up on the right. I've confused everybody. There we are. Yes, thank you.
We're coming to find now that glial cells, especially within the central nervous system, have a pretty substantial role in transaction of action potentials. Are you going to be using glia cultured alongside neurons on your diamond lattices?
That's a super great question. Yeah, so they've played they're playing an increasingly important role, as well as astrocytes as well. And so a lot of the neuroscientists are now culturing all of those things together to really mimic the environment of the brain. And in particular, so tonight I've talked about neuron cultures in 2 dimensions, where we have these networks cultured in 2 dimensions. The field of neuroscience is really evolving to do cultures in 3 dimensions, which is more, which capitulates the brain in a little bit a better way. And in those 3-dimensional cultures, absolutely, we've got glial cells and astrocytes to mimic the that environment, that 3-dimensional environment. What I didn't talk about tonight, I've got slides if you want to see, is work we're doing to make, and this is part of what Hunter and Elisa have been working on, to make probes that can go into these 3-dimensional brain organoids and actually start to probe activity inside these sorts of structures and tissues as well, which will be a really important advance, I think.
Okay, and I'm very conscious of the time, but I did promise that there was one more in here, if the person has the microphone.
Thank you. These are pretty quick. So appreciate the talk, very informative. You mentioned that the MRI scans are fairly expensive, but we also are talking about diamonds. So in the 5 years when we do commercialize this, can you give any thought to, what the differential might be? And also, rarely do we see these things only applying in one area. Are you starting to think about multiple applications of this technology?
Yeah, great questions. I don't know, Tad, can we get the slides up again? I wanted to highlight one thing I skipped over. So one of the challenges that in the demonstration that I didn't get to talk about was that we were using single crystal diamond, which is expensive. So the chips themselves are upward of $300 each. And if you wanted to do that on it, so there are 10 million iron tests that are administered in Australia every year. So it's not practical to do that using a single crystal diamond. The reason we went to nanodiamonds is they are super cheap. They are used, the main use of nanodiamond at the moment is for polishing parts. So they're done on an industrial scale, and I did a calculation. It costs 12 picocents for an individual nanodiamond in that solution. So they're incredibly cheap. We can buy buckets of nanodiamonds now for not a lot of money. The problem is we need to engineer that surface, so that's a really important consideration. That'll add to the development cost of the materials, but we think we're super competitive with the cost of a standard blood test now. So that's been one of the real driving factors of going to this. In terms of other applications, absolutely. So we're very good at detecting magnetic fields in using this, and there are lots of other paramagnetic things that you would want to detect in biology. So ferritin's a really important one, and it's a really great, there's already a need for that. But we are looking at other paramagnetic targets as well. So really great question.
Fantastic. Now, I know there was lots of other questions, but unfortunately, I'm going to have to draw it to a close. I'm very quickly going to hand each of you a little gift of our thanks. Don't forget that next week's lecture is all about quantum computing, and directly after this, for those of you in the room, we're moving outside up the bridge and into the Physics Museum for the reception. Thank you all very much, and please give these two a massive round of applause. Thank you.